we'll finish our conversation about functional genomics, and we left it at with new technologies. It's absolutely possible to sequence entire large genomes, like the human genome or any of the eukaryotic genomes, within a couple of days, right? Now, one thing that modern sequencing technologies brought about is the possibility of sequencing several genomes simultaneously as well. The idea is as follows. Bacterial genomes are approximately anywhere between 215,000 times smaller than eukaryotic genomes. So it's simple math. If in a couple of days you can sequence a eukaryotic genome, in a couple of days you should be able to sequence anywhere between 215,000 bacterial genomes. And that's what happens. Nowadays, you take any environmental sample, you subject it to these massive sequencing technologies, and you get these contigs in a way that end up representing individual prokaryotic genomes that were present in those samples, right? So now it's no longer an exercise of taking a sample, getting the genomic sequence, and annotating the one genome. It's about taking a sample, sequencing all of the genomes in it, and annotating them all, right? So we can move from sequencing one genome and annotating it to sequencing and annotating several. And that's something we call metagenomics. So the name implies that we're going beyond the genomics of one genome. We're looking at several genomes simultaneously, right? So this obviously applies mostly to microbiology, where the prokaryotic and viral genomes are going to be way smaller than eukaryotic ones. Just like when we annotate a large eukaryotic genome, we can make some predictions about what genes are expressed in those genomes. We can do the same for all of the genomes in a metagenomic collection, right? So not only do we get the genomes for all the species that were present in those samples, but we can also go into those genomes and start annotating genes as well. And in bacterial genomes, it's even easier because genes are intronless, there's a lot less intergenic sequences, everything is a lot more compact, and all you need to do is identify those strong promoters, open reading frames, ORFs, and then some strong terminator signal. That's it, that's your gene, and then on to the next, and so forth, right? So we can come up with gene predictions for all of the genomes. And that, like I was saying before, gives rise to metagenomics, which is essentially trying to start making predictions about the behavior of microbial communities. So we're not thinking about what a specific species could be doing with its genes. We can start thinking about what microbial communities could be doing with their collections of genes. Okay? All right. So how does this go? Typically, as I mentioned before, you take a sample that could be from soil, could be from your gut. There's a lot of research now being done on the intestinal microbiome. If you guys heard things like the gut-to-brain axis, and the story goes something like this: the bacteria in your gut produce a whole bunch of metabolites that are actually used by your brain. So whatever you have in your gut may be affecting your brain function. That's real. Samples from soil, your gut, an ice cube from Antarctica or permafrost. One of the faculty members in our department, Dr. McElprank, studies the microbes that are present in chunks of frozen soil that come from permafrost in the Arctic. It could be from contaminated waters. It could be from anywhere that you can think of and you have a question about. And one of the first questions that we can ask is simply, who is there? And what I mean by that is when you take these environmental samples, you can be pretty confident that you will find many of the organisms, bacterial organisms and viruses, that are known already, right? And you will be able to map some of the genomes that your sequencing project generated to a specific species. There may be some new genomes as well. There may be some genomes that computers are very confident are properly assembled, but then when you compare them to all the known genomes in databases, you can't find any that matches it. All that means is you've just found a new species, right? Or a new strain of a prokaryotic or viral species, right? So basically, trying to understand who's there, what bacterial and viral species are present in a sample, is what we sort of globally refer to as microbial ecology, right? As you guys probably know, ecology goes a little beyond simply listing the species present in an environment. It goes into trying to understand how they communicate with one another, and this is no exception. So microbial ecology is a little more than just a list of species. But oftentimes just being able to compare the not only presence, absence, but different relative proportions of different bacterial species in a community can tell you a few things about those samples. So for instance, going back to the idea of taking a look at the gut microbiome, the presence of certain bacteria in the microbiome may be indicative of physiological states, of predisposition to disease, maybe even sensitivity to certain drugs or toxins in the environment, etc. The way that's done is, here's obviously a cartoon, but you have your very complex collection of microbial species from whatever your sample is. You use next generation sequencing to generate the millions and millions and millions of short reads. And then you basically assemble these genomes based on that concept we discussed on Tuesday, shotgun sequencing, and the idea that with all of these millions of reads, you can start overlapping them and build the puzzle of each of these genomes. So with significant overlap among the millions of reads that you generate, you'll be able to assemble genomes A, B, C, D, and then, as I was suggesting before, you can contrast those genomes that you've assembled to databases of known species. And if you're lucky, you might be able to say, oh, look, I found Lactobacillus plantarum. Oh, look, I found Escherichia coli. Oh, I found so forth, right? And if you're even luckier, you might even stumble upon some new unknown species. Questions about that? That's pretty straightforward. Then we can take it a step further. As I was saying before, just listing the species can be informative in many ways, but it's often more fun to try and predict or estimate or make an educated guess as to what those species might be doing, right? So not only who's there, but what are they doing? And how do you think we can do this? After all, we're not really looking at these bacteria per se. We broke them up to extract the DNA out of them. So all of these bacteria are dead now. So how could we be predicting what they might be doing in those environments we found them? How would you do that? Excellent. So we're not looking at RNA, but you're right in the sense that if we have good gene models, gene predictions for each of those genomes, that means that we have coding sequences that we could translate, quote unquote. We call this in silico because we do it all in a computer using bioinformatics and super simple programs to take those operating frames that we identified in these genomes and ask the computer, can you please tell me what protein this would make? Now, translating in silico a nucleotide sequence will just give you the sequence of amino acids. That's not necessarily telling you what the protein does. But with a sequence of amino acids, and again, using pretty powerful computers, you can often predict what structures those polypeptides might acquire, how they might fold in space. And knowing the structures that they acquire in space, you might start making some predictions about what they might be doing within those cells biochemically, right? Now, I don't want to oversell the structure prediction. So structure prediction can tell you with a fair degree of confidence how a certain protein might fall in space. That extra step from structure predicting function comes really from knowing what we know about many other proteins that we know of. So I believe you mentioned the other day that sometimes when we're annotating genomes, you can look for sequences of known proteins in other species and see if we can find them in our new species, in our new genome. We kind of joked around with, I'm walking in the forest, I stumble a fungus, and it turns out that it's a new fungal species, so I can ask, does this fungal species have some of the proteins that I know of in other fungi, right? So same thing here. You can take some of those open reading frames, those gene predictions, in silico translate them to proteins, try and derive the structure from them, from that amino acid primary sequence, and then by analogy, or in some cases even homology to other known proteins in other organisms, you may start making some really very well-informed inferences about what those proteins might be doing. And a typical case in hand is enzymes with like weird catalytic capabilities, right? So there may be some microorganisms that have the ability to reduce methane, for instance. And those enzymes that reduce methane tend to have structures that are kind of unique. So those are the kinds of things that you could look for in new organisms that you know nothing about. You can start asking questions, well, can these bacteria reduce methane or fix nitrogen or do this or do that? Can they do photosynthesis, right? You can start asking those questions simply by asking whether or not they have proteins that may look like those that you know do those things in other organisms, okay? And then there is an even more fun question, and that is, what could they do? When you're doing this exercise of trying to figure out what certain proteins might be doing, sooner or later you're going to stumble upon cases in which the structure, the predicted structure of a protein that you just found, or a gene prediction that you just made in a given genome, is similar but not really identical to a protein that you know of in a different species. So now you can start literally creating and formulating all kinds of hypotheses as to what some of these new genes might be doing, right? So you may find that there is an enzyme that looks a lot, or a protein that looks a lot like one of those methane-reducing enzymes that you know about from other organisms, but not quite the same. So you can start making educated guesses about what those proteins could be doing. Maybe they're reducing something else. They're not reducing methane, they're reducing alcohol or something like that, right? And then another, I don't think I have a slide for that, but another fun thing that you can do in metagenomics is, again, take that microbial ecology approach. And if you know that there are certain organisms that produce methane in your sample and certain organisms that might be able to, well, you wouldn't reduce it, oxidize methane, then you might start, again, it's all hypothetical, but you may start making hypotheses about how these organisms depend on one another, or they thrive in the presence of each other, right? And that might lead to entire ecological webs of the kind that you would build with classical microbial ecology or multicellular ecology with big organisms, animals and trees, of, yeah, I can see how these organisms may be living off of these other ones, and these other ones produce a chemical that is critical for this third one, and the third one is recycling these molecules that are going to be used by the first, and next thing you know, you have this web of interactions, of ecological interactions between them. Okay? That's the kind of thinking that goes into, let's say, when people are trying to think about detoxification of highly contaminated water sources, or how could we deal with toxins in the environment. They go and they look in really harsh environments where they will find communities of bacteria that work with one another to get rid of really nasty stuff. And they try and learn from those communities by playing this game of, let's see who's out there, what they are doing to the best of our abilities, and even more so what they could be doing. So we try and start formulating some testable hypotheses that might lead us to the identification of certain key genes that then we could use in some type of biotechnological approach for bioremediation or detoxification using genetic engineering. We're now moving away from genomics, but I need to take a step back and bring us all back to the more classical genetics that we've been doing for the first part, the better part of the semester. Mendel's flowers, these mice that you cross to one another, flies. In each of these cases, we had a clear phenotype, right? And more often than not, we would carry out a forward genetics approach or a standard genetics approach and ask the question, what gene is behind these phenotypes? Right? This took decades back in the day, right? But eventually people started coming across these traits that were monogenic. So eye color in flies, this one locus in the X chromosome. Flower color, this one locus that Mendel found over 200 years ago. The B locus for mouse pigmentation. So all of these were monogenic traits, one locus behind these phenotypes. Cool. If we apply the same thinking to humans, there's been many cases in which it was just that simple. Human conditions or traits or diseases in some cases that are explained by one locus. So you take your human population, you split them between individuals who do not show or manifest a given condition, individuals who do, and then you compare their genomes, and it turns out that the difference between these two groups of individuals is really narrowed down to this one genetic sequence that is different between them. And there's many examples of monogenic human diseases. Right? I know that on Tuesday you asked me, or you suggested that maybe we could use genomics to ask sort of forward genetics questions, but much more quickly. I have these two groups of individuals, I collect their genomic information, compare them, and I should be able to see where the differences are, right? Like I have my group of control individuals who are not affected by a given condition or disease, my group of individuals who are, let's sequence their genomes, let's find the difference, right? It's a pretty straightforward approach. So essentially what we're saying is we have those two groups of individuals that differ in any given characteristic, like not having a disease or having it, and then we collect all of their genomes and let's use genomics approaches to find where the differences are. The question becomes, where do you look for differences between these two groups, right? Because when you have these genomic sequences, you're going to have, if you will, a book with 3 billion letters in each of these books for every person that you sequence. Right? So where do you look for differences? That's the question, right? So the exercise that I wanted to run with you is the following. Let's say that we home in on these 3 different areas of the genome. And again, the scenario here is there is a group of individuals who are not affected by a disease and a group of individuals who are. So my question to you guys, and you can work in groups, which of these 3 sequences would you use to determine what the genetic difference causing this disease might be? So let's go bit by bit. You said it. They all look the same. So when there is absolutely no difference across individuals, that's simply a not informative area of the genome. And there is plenty of areas like this in our genomes where all of our genomes are identical because they make code for critical proteins that have been selected to have a specific sequence, and it's very hard to move away from those sequences. So those are ultra conserved regions, and those sequences are going to be identical across all of us, and chimps, and maybe even some other mammalians, right? Cool. So first column, not very informative because they're all the same. Now we get into a pickle, right? Second or third column? What do you guys think? Yes.
I would say the third column.
You would say the third.
Just because while the second column does have the starting codon, like it has the TAG, which when it goes through transcription, it'll have the AUG for translation. But so does the third one, and the third one also has differences in the code, so we can see like
differences in the code. Okay, so I have someone advocating for the third column because there are more differences. Is that okay if I paraphrase it that way? There is more differences in those sequences. Okay, cool. Yes.
I was thinking the second one, if you're looking for like a monogenic trait, you want to look for something that's like a more specific difference, like the C and the G in the fifth letter of the second.
Okay, so you, like I'm sure many others, notice that there is that position here. There is G for about half of the sequences and C for the other half, right? Okay. Any other? So we have these two competing ideas. One is there's more diversity in the third column. The second column is better because there is a more specific difference between the sequences. Any other arguments for or against either column that you want to share, maybe from your conversations? All right, so let's talk about these two ideas. So what the second column has going for it, if I understood you correctly, is that there is one difference. So it's a lot more specific. We're looking for a monogenic condition, hopefully, cross fingers, right, that is monogenic. So if the condition is monogenic, there's a good chance that the change is in one gene. But are you looking at a difference in one gene here or one letter? Right? It's only one letter. We don't know if it's a gene, not a gene, in the coding region of a gene or not in the coding region of a gene. It's just sequences. I appreciate the fact that you were looking for the ATG, but remember, when we're doing genomics and in general when we're comparing sequences, we're not always focusing on protein coding genes or the beginning of a protein coding gene where you might look for the ATG/AUG. You could be looking anywhere, right? So, but let's not let go of column 2 yet. What's the advantage of column 3? More options, more diversity, right? More differences. So it may be, column 3 may be better to detect changes that are associated with not having the condition or having it. But that degree of diversity brings about a problem too. You guys don't know this? I do because I built these things. But these sequences are all different from one another. There's not two that are the same. So it can get a little tricky to determine what features, what combination of letters within these sequences might be associated with having the condition or not, right? It can get a little tricky because who's to say that it's the 4th position or the 8th or the 12th? We just don't know. It's so all over the place, right? In fact, by design, all of these sequences are different to kind of emphasize. I could have hidden like a letter that is shared by, no, they're all the same. I mean, all different. There's no association whatsoever between a given position and letter and having the condition or not. So it turns out that when we're looking for differences that might allow us to group individuals in, let's say, these two groups, that middle column is about the best approach. And the reason is that there is a way to clearly group the sequences in two classes. the ones with the G, the ones with the C. Now, does that mean that the ones with the G are going to be no condition and the ones with the C, the one with condition? Well, not necessarily, but at least if they were, we're a lot closer to saying, I think there's something here, right? So what we're looking for, if we're splitting the population into two groups, we're looking for sequences that might allow us to split the genomes into two. And then we see what degree of overlap there is between the two genetic groups or genomic groups and the two biological groups, having the condition or not. Does that make sense? Yeah? So this idea of looking for regions of the genome that are almost identical except for one position gives rise to a concept that is used a lot in not only medical genetics, but also evolutionary biology, studies of ancestry, forensics, and it's the concept of the single nucleotide polymorphism. We kind of touched on this before, the idea that individuals in a population may differ in one very detailed aspect of their genetic sequence in a way that might allow us to say, okay, now I'm a little more confident that we're not the same when it comes to that middle column. We're not the same when it comes to that third column either, right? If we were to use the third column to compare us all here, we would probably get all kinds of different sequences, right? If we were to use the first column to compare us all in this room, we would all be identical. So we need something that allows us to group us into groups that are not equal to the number of individuals in the set, right? If I were to split us all in, class has 70 students, let's say there's 60 of you today here, plus me, 61. If we end up with 61 groups, we didn't really do much. We started with 61, we ended up with 61, no progress. Does that make sense? So the single nucleotide polymorphism is a concept that became very, very handy because it's basically saying, yes, there is these regions of the genome that are ultra-conserved except for this one position. And if I know what letter different individuals in a population have in that position, I know to which morph they belong, and since the term single nucleotide polymorphism, because there is various morphs or various forms for that, right? So the SNP. If we were to translate or somehow depict those columns as color boxes, the first column would be same color for all boxes. I appreciate that it's not coming through all that nicely. In the slides, it looks a little better, I think. The second column, there is only two colors to them, either magenta or cyan. And then the last column, all kinds of colors, different colors, because these sequences are all different from one another. Okay? So if we can home in on an SNP that hopefully has only 2 or 3 classes, then we run that exercise of seeing how much these 2 classes overlap with these 2 classes. And if we were to find that all of the genomic sequences with a blue box in the second position are the healthy individuals, and all of the individuals with the magenta box are the diseased individuals, bingo, we found something, right? Cool. So ideally, we were hoping for a monogenic disease, and we were hoping for an SNP, hopefully, that would allow us to 100% distinguish between the non-condition and the condition group. In reality, when we do this genomic sequencing and we look for SNPs, this is more like what we find, right? So to your idea of let's just compare the genomes and see where the difference is, it's very hard to pinpoint the one position that is different between the individuals. Not only there's going to be many locations at which there are differences across individuals, but also the degree of overlap between one variant or the other, one genetic variant or the other, and the biological phenomenon is not 100%. Yeah? Did that make sense? I'm going to ask you to run another exercise. Now looking at these genomic sequences on the right side of the slide. And if you have a neighbor with the slide on their screens, you may be able to see it better. Which of these positions might correlate better with having the condition or not? We'll call that position number 1, 2, 3, 4, 5, 6, 7, 8. That's 10. Which of those SNPs correlate best with the condition? So what do you guys think? Any candidates? 9. I hear 9. And I heard a 2. Yay! Those two are the ones that it's not 100% correlation, it's not a perfect match, but both 2 and 9 show an enrichment for blue flavor among the non-diseased individuals and magenta or pink flavor for the diseased ones. Right? So what is that telling us? It's just telling us that those particular SNPs correlate a little better with the condition in question. Meaning that if an SNP is of a flavor, there's a higher chance that you either have or not the condition, right? So if that second SNP is blue, then there's a higher chance that you're fine for that condition. If that 9th SNP is blue, then you're probably fine for that condition. If either or both of the 2nd and 9th SNPs are the pink flavor, then you may be more predisposed to having the condition, right? That is, what I just described is what we normally call a genome-wide association study. You are associating the correlation between different, typically SNPs that are widespread throughout the entire genome, and you do it genome-wide. You don't do it for a specific location in a given chromosome. You do it across all chromosomes, across all chromosomal positions. So genome-wide association studies. And for short, people refer to this as GWASs. And you encounter in the literature, anytime that you read these days, a genetic study found that this or that gene is associated with condition X. It could be, a physiological condition, it could be a behavioral trait, it could be anything. Chances are that behind that statement, there is a GWAS. Someone took the many genomic sequences that are available these days in public databases. For each of those, there is clinical histories associated to those genomic sequences, so you know the individuals behind those genomic sequences, whether they experienced any conditions, they had any trait of interest, even things about life history, habits, diet, etc., etc., etc. And then you can do one of these GWASs, and what you do is you look for those SNPs or those loci across the genome that associate most with the condition. What I mean by that is if you have one flavor, you're more likely to have the condition. If you have the other flavor, you're more likely not to. Did that make sense? GWAS. There's a very peculiar way in which you see some of these things depicted. They're called Manhattan plots. And there is a chance that at some point you come across them in the literature. So a Manhattan plot, a classic Manhattan plot, will have the chromosomes on the x-axis. So you'll notice the numbers from 1 to 22. So there's one that is missing there. We don't know which one. I'm guessing the sex chromosome. So 1 to 22, so this must be the somatic chromosomes. And then on the y-axis, you depict or you plot the negative logarithm of the probability of association. So whenever we say that a given locus is tightly associated with the condition, that means that the probability that we're making a mistake in making that claim is really, really low. So anytime that you find something that is statistically significant, you will find things that are numbers that are, p equals 3 times 10 to the minus 7 or something like that, really small numbers. So those really small numbers are kind of hard to graph. So what we do is that negative logarithm of it. So the negative logarithm of 3 times 10 to the minus 7, it approximates to 7. Right? Like pH, if you guys remember your pH and POH days. So now we can plot the p-values of each of the S&Ps, and in this silly example, we could imagine that what we drew here, and there's not much whatsoever, this is just it's a cartoon for learning purposes, but we can imagine that this chromosome is actually chromosome 3, and that these two positions here that correspond to S&Ps 2 and 9 have really high negative logarithms of the p-value, so really high correlation scores. Right? Did that make sense? Now, what do we do with this information? How do we take it to the next step? Because these sequences that I showed you before, and as I stressed, I think pretty strongly, they don't have to necessarily be protein coding sequences. They can be sequences that exist anywhere in the human genome, for which there is that sweet balance between having a lot of conservation except for one position. Those are the sweet sequences that allow us to run these comparisons, right? That make it very binomial. Either you have it or you don't. If you don't do that and you go for positions that are more complex, the math gets way more complex. But this way is easy. Either you have it one flavor or the other, then you have the condition or not, and the correlation studies get easier. Now, if these SNPs don't exist, let's say in coding sequence, if you go into those SNPs and you look around and it's not within a gene, what do you do with that information? How can you link an SNP that exists in the middle of proverbial nowhere in the genome to a disease? Shouldn't it be in a gene? So how do we use this to try and find what the actual gene behind the condition might be? I come to you and I say, look, this SNP has a super strong correlation with this condition, but it's not in a gene. Can you help me? What might you try? Exactly. Wasn't that intuitive? You just look around on both sides of this thing, right? You look around. Now, the SNP is here, right? And the SNP, for lack of a better term, is tracking with the condition, but the SNP is not causing the condition. So why looking nearby that SNP might help you in finding the one gene that is truly causing that condition? Because of something that gave you a little stress a few weeks ago, but now it's coming all together because of linkage. Oh. Remember linkage? When things were really close to one another in DNA, that they never separate over very long evolutionary timescales? So the idea here is if these SNPs were traveling one way or another into non-disease individuals or diseased individuals, there is an extremely high chance that anything that is really close to those SNPs traveled with them. And the gene that is causing this disease but doesn't have an SNP necessarily associated with it, you didn't detect it because the mutation causing this thing doesn't classify as an SNP. It can be a large deletion, it could be anything else. So it doesn't count for tracking purposes, and yet it's going everywhere where these tracking SNPs go. So these SNPs don't point us directly to the genes that are causing certain conditions, but we can identify them through a concept, and I kid you not, it's a concept referred to as GBA in genomics, and it stands for guilty by association. Guilty by association. Right? So you will find in literature that SNP 23.5AX was highly correlated with diabetes, and genes A, B, and C were identified as potential candidates, as potential GBA candidates. And what that is telling you is that those genes were in the vicinity of these SNPs. So linkage at some point, and I do remember, I remember because it's easier for me, but I remember bringing this up when we talked about linkage. That linkage was a fun thing that geneticists did close to 100 years ago to map genes based on recombination frequencies, right? The less two genes or two alleles recombine with each other, the closer they were, and people build these maps. Oh, fun. Who cares about these maps anymore, these genetic maps? Now we sequence the whole thing and we're done. But linkage is still very, very useful as a concept, because now we find these SNPs through GWAS associated to a given condition, and immediately we go and look in the vicinity of them, because we know that through linkage, if those SNPs were tracking with a certain condition, there's a good chance that neighbors would track with the condition as well. Yeah? So how these happens in real life, you hear someone doing a GWAS, they'll first identify the SNPs, and typically what they will say is, we found 20 loci within 0.5 centimorgans of those SNPs, and that's already telling you, yeah, very close, very low chance of recombination, so it's a good chance that these loci, at least one of them, might be associated with the condition you're interested in. Okay? That's one point that I wanted to make. And another point that I wanted to make is I presented this case in a very rosy way, right? Like you do the GWAS and you find two candidate loci within one chromosome. The reality of GWASs, when you actually start looking at many of these Manhattan plots, looks a little more like this. What is that telling you? Is it monogenic? Probably not, right? I see at least 10 different loci that associate tightly with the condition. So that means there's at least 10 loci that may be contributing to this condition. That means it's not monogenic, right? And that is the reality of most GWASs for a good reason. Most of the human monogenic diseases, I don't want to say they've all been discovered, but many of them have, because you can get to them rather quickly using old techniques, pedigree analyses, right? And then you do some karyotyping and FISH, some of the things that we discussed in this class, and then you can relatively quickly get to the area of the chromosome that may be associated with the condition, and then you home in on the gene. GWASs nowadays are normally run for much more complex conditions, things that we know are not monogenic. So you run a GWAS for things like, again, these are examples, but cardiovascular disease or diabetes or rheumatoid arthritis or psychosis, like these very complex conditions that we know are not monogenic. If they were monogenic, they would track easily in a family pedigree. We would know it, right? These are way more complex. And what we're trying to do is, fine, if a GWAS can take us to the 50 loci that track most with the condition, well, we reduce the problem from 20,000 genes or 24,000 genes to 50. That's progress, right? Did that make sense? So for illustration purposes, I showed you that very simplistic Manhattan plot at first, but most Manhattan plots from GWASs will look like this, with many positions across all chromosomes tracking with the condition of interest. Okay? All right. And for the grand finale, an application that is not so much associated with medicine and medical genetics of GWASs is ancestry analysis, right? So up until a couple of years ago, I would immediately say 23andMe. Now they went bankrupt. In case you didn't know, they went bankrupt, so they are a business. The sequences they were not trash, so there will be stored forever, but the company itself went down. But ancestry analysis is based on this same principle of GWAS. The condition, quote unquote, is simply geographical origin, right? And the idea here is that these companies homed in on groups of SNPs that tracked very, very tightly with very localized geographic locations. So they went into all continents and they homed in on communities where there had been very little mating with populations from other places on Earth, right? So they went into, say, Australian tribes and South American tribes and African tribes, Asian, Europe, everything, right? And then you come up with these groups of SNPs that track with certain geographic locations. And when you grab an individual who submits the spit on the tube to find out what their ancestry is, what you look for is combinations of SNPs, and this translates kind of linearly to that famous, you're 50% Northern European with, 30% South American and 20% this, and right? And the idea here is that in your individual history, in your ancestry, matings between individuals from different places on Earth brought together different combinations of these SNPs. Okay? So it's based on the same principle that certain SNPs track very, very tightly with a given geographic origin, and then all you do after that is ask what the quote-unquote flavor of those SNPs is. I want to point out, it's not like this position does not exist in individuals from other geographic locations. I'm not implying that. What I'm saying is that that particular flavor of that SNP tracks with Northern Europe, and this particular flavor of this SNP tracks with South America and so forth, right? So everyone will have those SNP positions, just the specific variety or the specific type that often associates tightly with a given geographic origin. And the same applies to diseases, by the way, I should point out. So I made it simplistic, saying the blue box or the pink box. For an SNP, the blue box could be that, remember that C and G that you noticed? That nucleotide that was different for that particular SNP? It could be that blue is having a G there, and the pink is having anything but a G, right? So these binary distinctions that we make of SNPs sometimes go down to having one letter at that position is one flavor, having any of the other three letters is the other. And I just wanted to close with this. So, last two slides of the semester, and leaving you with the idea that, we talked about flowers and mice, but towards the end of the semester, we started getting a little more serious about how we use all of these things. So this is a map of genomic companies that do different things. It's slightly outdated. This is from 2020, I think, or 18. So you have direct-to-consumer kits. 23andMe is still there, no more, but some of the other ones that you may know, Ancestry. You have carrier screening genetics risk companies. So these are companies that will take a sample, typically saliva sample, and then they can figure out if you carry any alleles that are known to be associated with diseases. Companies for non-human genomics software. Tumor profiling and cancer treatment guidance. I do want to spend a minute on this because I know that it's mentioned in one of the previous slides, but we didn't really go deep into it. There's this new thing now called pharmacogenomics. And what that means is when researchers, mostly companies like this, are interested in trying to understand if there are any genetic differences, and by extension, genomic differences, that might explain why certain people respond well to a drug and others don't. More often than not, when a company produces a new drug, looks awesome with cells in culture, looks amazing with mice, looks great in clinical trial phase 1, phase 2, then they release it into the general population or phase 3, with a larger population, and now all of a sudden it doesn't do anything. Why? It's not because it's not helping everybody, it's because it's helping a lot, a group of individuals, and the rest it doesn't do anything. Right? The drug fails, and that drug, even though it may or may not get approval, the company won't produce it because they can't make any money out of it. About 10 years ago, this thing exploded. Pharmacogenomics, personalized medicine. You may have heard that term before. Personalized medicine, pharmacogenomics. The idea that more and more we're understanding how certain drugs may be good for you or not, may be good for your particular type of tumor or not, depending on what your genetic sequence is or what genetic mutations that tumor that you have has. Right? So there may be tumors that respond to a drug or not, depending on what mutations they gave rise to them. So now there are company after company after company that will take a sample of your tumor, obtain a genome sequence of that tumor, and use that information and everything that we've learned so far in pharmacogenomics to let the doctor know and let them make a more informed decision whether or not they should give you drug A or B. Because it turns out that studies show that with that particular genetic variant in the tumor, drug A does not work. Right? So pharmacy, genomics. Early disease detection, clinical research, databases, tools, etc. So huge market. And of course, this has exploded even further with the advent of AI. It used to be that you had to have in-house pretty skillful mathematicians and statisticians to deal with all of these. Now, theoretically, these companies will tell you that just give me the genomic sequences, I'll feed it into an AI, and the AI will tell me what SNPs I should be looking at, why, how they correlate with certain drugs or not, and so forth. So there is a market out there, and obviously this class won't do it for you. It will require further training, but it's not just if by any chance you were thinking, oh, I'm taking genetics, I'll just learn about Mendel's flowers and whatever. No, there is actually career opportunities in genetics, and probably more so now than when I started in biology, because when I started in biology, there was Sanger sequencing, but no capillary sequencing.